Hello everyone, my name is Sarah Saxton. I'm a certified genetic counselor at the New York City Office of Chief Medical Examiner. Today I'll be presenting postmortem genetic testing results and how to communicate them. During this talk, I will provide a simple genetics background, which will facilitate how to understand a genetic test report, and at the end I will talk about how I communicate postmortem genetic test results to the families of our decedents. My role as a genetic counselor here at the New York City OCME is unique as we are the only ME office in the nation to have an in-house genetic counselor. I am involved in interpreting genetic test results, writing up reports, and determining whether genetics were involved in the cause of death. This is often a very collaborative process between myself, Dr. Tang, our geneticist, and the ME involved in the case. After the ME first discloses the results to the family, along with other results and findings, I personally speak with families in more detail about the results, their family history, and make recommendations based on what we discussed. We do genetic testing because it provides answers for the cause of death, which is important for not only the certification of death, but it can also provide more closure to the family as to how their loved one died. Determining a genetic cause of death can also inform family to their risk, as we know blood relatives share genetics. Many of the conditions we test for present with few or minor symptoms prior to a sudden death. Therefore, genetic testing can alert family and they can initiate early medical intervention to prevent another sudden death from occurring in the family. Genetic testing, very simply, is a tool that allows us to read our genetic code. Our genetic code is essentially the instruction manual to how our bodies form and function. When we do genetic testing, we are searching for genetic differences, or what we call variants. We expect to see genetic differences or variants in everyone, as part of what makes us all different. However, when we do postmortem genetic testing, we are specifically looking for genetic differences that may explain the cause of death. Sorting which of these differences are part of a benign population variation and which are disease-causing can be a challenging process. Genes are packaged together on chromosomes. Chromosomes come in pairs, one from mom and one from dad, which means for most genes in our bodies we have 2 copies. The major exception is for our 23rd pair of chromosomes, the sex chromosomes, where females have 2 X chromosomes and therefore have 2 copies for each gene on the X chromosome. However, males have an X and Y chromosome, which have different composition of genes. It can be helpful to think of our genetic code as an instruction manual with chromosomes representing chapters and individual genes being equal to a single instruction. If there is a disruption that changes the instruction manual in any way, it may or may not cause the affected instructions to not function anymore. The testing we do at the OCME concentrates on looking at specific single genes associated with sudden death. Dr. Tang's talk goes into more detail about the genes on our panels. When we see there's a difference in a gene that is expected to make the gene not work properly, it can lead to a sudden death condition. It is important to be aware that our genetic testing method detects small spelling changes in the genome. Other changes, such as changing the order or duplicating genes, may not be detected by our testing methods. Genetics is also a rapidly evolving field, and we may not be aware of all the genes we should be looking at for those high risk for sudden death. While this talk does concentrate on genetics, I think it's important to remember that genetics is not the only thing that determines who develops a sudden death condition. Environmental factors such as drug use, unhealthy diet, smoking can influence the likelihood of someone developing a number of health conditions. In combination with genetic factors, this may increase someone's risk even more. However, the genes we test for when they do not work properly greatly increase the likelihood of developing a sudden death condition, regardless of any environmental factors a decedent may have had. Based on the autopsy and available records alone, it may be challenging to determine who has a genetic predisposition. In our experience, decedents who appear to have clear environmental factors for the cause of death, such as hypertensive cardiovascular disease and cardiomyopathy, have tested positive for a genetic cause. Therefore, genetic testing may be justified to rule out a high genetic risk for surviving family members. Because sorting out which variants cause disease and which don't can be a complicated process, having as much of the following information is crucial for labs to determine which variants to report. Be sure to provide a decedent's relevant autopsy findings, medical history, dysmorphic features that may be suggestive of a syndromic condition, and any reported family history of sudden deaths or signs/symptoms of the suspected condition. Moving into how to read a genetic test report when a variant is detected. This is the basic information that every genetic test report should have in order to determine how genetics may have played a role in a decedent's death. A report should include the variant's name, what gene it was found in, the conditions and inheritance associated with the gene and/or variant, the zygosity, or how many copies of a variant were detected, and the lab's classification of the variant. This table is an example of our summary table for our genetic test results. I'm going to discuss types of inheritance patterns, zygosity, and the classification of variants. Again, the genes and their associated conditions have been covered in more detail by Dr. Tang. Start off, a variant can be classified into one of three main categories, either benign, a variant of uncertain significance, or pathogenic/likely pathogenic. When all the variants detected in a test are considered benign, the report will state negative results. This means no disease-causing variants were found, and this test could not provide a genetic explanation for the autopsy findings and/or cause of death. This does not eliminate the possibility of genetics being involved in the decedent's death, and the family may still be considered at an increased risk because of the decedent's medical history, including autopsy findings, or other family history. Clinical evaluations and surveillance should be recommended based on family history. As a reminder, every genetic test has limitations and may not be able to detect a genetic predisposition. We do next-generation sequencing, which looks for small spelling differences in instructions related to cardiac, seizure disorders, and other sudden death conditions in a decedent's instruction manual. Other methods, such as whole genome sequencing, look for genetic differences in the entire instruction manual. When a variant is detected, it may be called a variant of uncertain significance or a VUS. In this case, the lab found a variant but does not have enough information to determine whether it causes disease or how it impacted the decedent. Again, the family may still be at an increased risk for a sudden death condition from the detected VUS variant or another genetic cause undetected by the test. The VUS results themselves should not be used to guide clinical genetic testing or care because it's unclear whether it would cause disease in any family member that tests positive. That being said, additional genetic testing and clinical evaluations for other family members may give a bigger picture of how the variant impacts the family, and a clinic or lab may use that evidence to determine whether or not it's disease-causing. A variant may be classified as pathogenic or likely pathogenic, which means there is sufficient evidence to determine the variant is or likely is disease-causing. If the variant corresponds to the decedent's medical history or autopsy findings, it may be determined to be involved in the cause of death. A variant may also be considered a secondary finding, meaning it is disease-causing, but the disease was not observed in the decedent and was not involved in the cause of death. In both of these scenarios, it is important to alert the family of the variant since it can be used to guide their medical surveillance and intervention. It is the hope that armed with this genetic information, family members can learn who is at an increased risk of sudden death and get earlier medical care than they would have otherwise, thereby avoiding preventable deaths in the family. Even though pathogenic variants are known to be disease-causing, the likelihood of someone with that variant developing the associated disease depends on several factors. Penetrance is the degree a variant influences the development of disease. When a variant is fully penetrant, it is essentially guaranteed that a person will develop the associated condition at some point during their lifetime. Whereas if a variant is reported to have reduced penetrance, a person carrying the variant is not guaranteed to develop the associated condition. Variable expressivity is when a variant presents itself differently in different individuals, which may make it challenging to predict who will develop life-threatening features of a variant. For example, we see this with our aortopathy panel, which has several connective tissue disorders that affect many body systems, but from a sudden death perspective, we are most concerned about the aorta rupturing. A variant is considered de novo when it is new to an individual and was not inherited from either parent, but they, the decedent, can pass down the variant to their children who would be at an increased risk. It is also important to consider the inheritance pattern associated with a pathogenic variant. Three main types that I will be covering are autosomal dominant, autosomal recessive, and X-linked. In autosomal dominant inheritance, if we remember, we have two copies of every gene, one from mom and one from dad. A person that has one pathogenic variant on one of the copies is enough to develop the disease, which is also known as heterozygous. This means that when a parent with a pathogenic variant passes down one copy of their genes, they either pass down their functional copy or their non-functional copy. Therefore, there is a 50% chance their children will carry the pathogenic variant. This is the most common type of an inheritance we see at the OCME. If we expand this to think about the extended family, we have here a decedent who has tested positive for a pathogenic variant. Before testing relatives, the chance that any of the decedent's first-degree relatives, parents, siblings, and children would also have a 50% chance of carrying the pathogenic variant. And their second-degree relatives, grandparents, aunts, uncles, grandchildren, etc., would have a 25% chance of carrying the pathogenic variant. And that risk continues on to more distant relatives. Everyone has their own threshold for what they consider high risk. However, when we are talking about sudden death conditions, I think most people would be concerned about these odds. Testing more closely related blood relatives can help determine who in the more extended family to test. For example, in this scenario, both mom and dad of the decedent got testing. Because dad tested positive for the variant, we now know the decedent inherited it from him. It now shifts the likelihood of other family members carrying the variant, so that dad's side of the family is at an increased risk, whereas because the mother of the decedent was negative, her side of the family does not need to do genetic testing. Parents, after finding out their child died of something genetic, have expressed they would feel too guilty finding out they gave the variant to their child, which ultimately led to their death, and are therefore resistant to getting genetic testing for themselves. I explained no one controls what genetics they pass down or what genetics they receive, and the intention of postmortem genetic testing is not to establish who is to blame for their child's death, but to determine who would most benefit from early medical intervention. It's ultimately the person's decision to decide whether or not they want to know if they have a pathogenic variant, but we do recommend sharing the results with other family members for them to make the decision for themselves. Another type of inheritance we may see is called autosomal recessive inheritance. In this pattern, a person may carry a pathogenic variant and be asymptomatic, but when they have two pathogenic variants, they are at an increased risk to manifest disease. This can occur when the two pathogenic variants are the same, also known as homozygous, or when the two copies of a gene are different and pathogenic. We typically see this when two parents are carriers or heterozygous for a variant, and they both pass down their non-working copies of a gene to their child. Statistically, their children have a 25% chance of inheriting both non-working copies. It is rare in the general population to be carriers for the conditions we test for, but families may want to get testing for themselves and their partners to determine who is at an increased risk for developing the disease. The last inheritance pattern we are going to talk about is X-linked. Because males and females have different sex chromosomes with a different set of genes on them, the likelihood of an individual with a pathogenic variant developing a disease is dependent on their sex. A female can have a pathogenic variant and have less severe condition or be symptomless because she has another copy of the gene on her other X chromosome. However, males with their single X chromosome will be at risk of developing disease if they have a pathogenic variant. In these families, we are often most concerned about identifying females who may be carriers and their sons who have a 50% chance of inheriting the pathogenic variant. So as a summary of what we've talked about, once we identify a pathogenic or likely pathogenic variant, it determines medical management for family. We recommend family members get genetic testing through a clinical genetics program, but if those are not accessible, they can also have their primary care physician order testing, or some labs allow individuals to order genetic testing directly through them. With pathogenic test results, their medical surveillance and management can be based on a variant-associated inheritance pattern, predicted penetrance, de novo status, and expressivity. Identifying a pathogenic or likely pathogenic variant is much more likely to get covered by insurance in these cases. When a family gets negative or VUS results, they may still be at an increased risk to develop a sudden death condition. The family may benefit from clinical evaluations and more extensive genetic testing to further determine their personalized and family risk. If more extensive genetic testing is warranted, testing an affected individual, usually the decedent in our cases, is most beneficial, and in our lab we can send out samples to other labs for further testing. It is important to note that with negative or VUS results, a family may have challenges getting coverage for their own genetic testing or additional testing for the decedent. However, a clinical genetics program may be able to help a family navigate those challenges. In this next section, I will discuss 3 examples to illustrate how I talk to our families about the genetic test results. This broadly breaks down into 5 main points. 1. What is genetic testing and why was it done? 2. How genetics contributed to the decedent's autopsy findings and cause of death. 3. What it means for the surviving family members, including asking about additional family history. 4. Recommendations for how the family can move forward with these test results. And 5. Addressing a family's emotional, financial, health, or other concerns. I was talking to a decedent's mother about her 23-year-old son who had myocarditis and a history of fainting. I explained we did genetic testing because there are reports of people with myocarditis having poor outcomes like sudden death if they also have a genetic difference that impacts their heart muscle function. In this case, we detected a PKP2 pathogenic variant that causes autosomal dominant cardiomyopathy. Because this variant was detected, there is a 50% chance other family members will also carry the variant. And when we were discussing their family history, his mother noted there are 4 paternal relatives who died suddenly. Unfortunately, the first sign of cardiomyopathy may be sudden death. Therefore, identifying them prior to a fatal outcome is crucial for treatment. Genetic testing and cardiovascular evaluations are strongly recommended for first-degree relatives of the decedent, and other family members, especially on the father's side, may also want to consider genetic testing. Those who are negative for the variant would be considered low risk and not need additional follow-up, whereas those who are positive would benefit from regular cardiovascular screening to ensure proactive intervention, such as medication or ICD placement. His mother was grateful to have answers for her son's passing and options to prevent another sudden death from occurring in the family. The next example involves Marfan syndrome, which is caused by variants in the FBN1 gene. It's an autosomal dominant connective tissue disorder that can affect a variety of body systems, most notably the ocular, skeletal, and cardiovascular systems. From a postmortem perspective, we are most concerned about ruptured thoracic aortic aneurysms and dissections, mitral valve prolapse, and cardiomyopathy. It is a condition with variable expressivity, so not everyone with Marfan syndrome will develop features associated with sudden death or this lanky body type that is often a flag to assess someone for connective tissue disorders. In this case, the 33-year-old decedent had a known clinical diagnosis of Marfan syndrome, but never did genetic testing. For this case, we confirmed the decedent, who died of complications from Marfan syndrome, had a pathogenic variant in the FBN1 gene. When speaking with his mother and brother, they confirmed that first-degree relatives had been clinically evaluated for Marfan syndrome when the decedent was younger, and they were negative. However, at this time, they could not afford genetic testing. We recommended they go to a genetics clinic familiar with Marfan syndrome for testing and evaluation, determine if this variant was a case of the variant being de novo or variable expressivity with other family members with the variant not meeting clinical criteria for diagnosis. Now that they have the results and genetic testing is more available, most insurances should cover their genetic testing. And the brother expressed relief for finally being able to confirm whether or not he did have Marfan syndrome. In this final example, we had a 21-year-old female who was found unresponsive after a low-speed car collision. She had a lot of features consistent with connective tissue disorders like mitral valve prolapse and pectus abnormality, but was never diagnosed with a specific syndrome. The ME ordered cardiac and aortopathy panels for this case. Her cardiac panel came back negative, and we did find a VOS on the aortopathy panel in the PRKG1 gene. This gene is associated with aortic aneurysms and some features of connective tissue disorders like pectus abnormalities. However, it is unclear whether this variant was responsible for the connective tissue features in the decedent or if it was related to her cause of death, especially because she did not die of an aortic rupture. When I talked to the decedent's mother, she explained that none of the decedent's first-degree relatives had connective tissue disorder features, but there was a cousin who had a connective tissue disorder called Ehlers-Danlos syndrome. She was unsure whether the cousin had genetic testing to confirm, but we discussed how the cousin's results could be helpful since we test for a type of EDS associated with sudden death and not other types. The decedent's mother felt guilty for her daughter's sudden passing, but I reassured her that she had made sure she kept up with her medical appointments and advocated for her as much as she could. Unfortunately, our lab was not able to determine a genetic cause of death for this decedent. However, her first-degree relatives should get cardiovascular evaluations based on the decedent's history, and they may consider additional genetic testing. Finally, with families, I like to share local NYC resources for families to consider genetic testing through. Families can get testing through their primary care physician, but not all doctors have experience at guiding patients through the process or interpreting results. We have two main lists for cardiogenetics and epilepsy clinics listed here. It is important to emphasize to families that they need to bring the test results with them to the genetics appointment to ensure the clinic orders a test that will detect the same variant as the postmortem testing. That brings me to the end of this presentation. I hope you can appreciate that postmortem genetic testing can save lives after a tragic loss of a loved one. Of course, this wouldn't be possible without the hard work of our dedicated molecular genetics lab team here. And if you have any questions or would like to contact me, feel free to reach out to my email here. Thank you.